Thank you very much. I'm really pleased to introduce Outpace Bio. Our technology comes out of David Baker's lab at the University of Washington, where my co-founder and I took the field of protein design from the ability to make proteins with the right shape to make completely new biological functions. And we spent 2 years at a company that we co-founded called Lyell Immunopharma, where we were really taking this technology into solid tumors to really make a big difference there. Spun out from there to make Outpace, and I'm really pleased to tell you about it today. There are 6 approved CAR T-cell products for anti-cancer applications. They've shown an incredible potential to cure patients with a small proportion of heme malignancies. These are patients who had months to live and no other therapeutic options. And this is a piece of what has made cell and gene therapies such an exciting space with the potential to cure the cancer and other diseases of our time. There's been an incredible amount of investment in the space. A lot of that has been allocated towards building industrial scale. But here's the thing, the technologies that we've applied to these first indications are only going to be able to address less than 5% of cancers. And the problem is biological. Our cells, our immune cells, are incredible. They have polyfunctionality, they're able to expand, they're able to coordinate response from the rest of the immune system. But our immune systems have evolved in the context of viral infections, and they've evolved in the context of avoiding autoimmunity. And the mechanisms that our immune system has developed in order to make sure that we can survive in those contexts are exactly what cancer uses in order to turn off immune responses. And so, and this biology is deeply ingrained in how our immune systems function. It's really important that it's really robust. And so we have the tumor that's able to suppress an immune response. We have our immune cells that intrinsically turn themselves off when they chronically see an antigen. And the engineering tools that we brought to the challenge in order to address these problems are really limited by the existing biology that nature has created for us. So imagine making your computer or making your cell phone from parts that you go out in the woods and pick parts off of trees. So what we're doing at Outpace is using protein design to create the biological units of function that are going to address all of these problems. And one of the special things about cells is that you can put multiple solutions into the same cell in order to address all these mechanisms at the same time, which we know we're going to need to do if we want to cure these the rest of the 95% of cancer indications. And I just wanted to start up front with a little bit of data just to show you how much progress we've made even in this short amount of time that we've had. For some of you who aren't used to looking at this data, this is a xenograft solid tumor model where you take tumor cells and implant it on an immunocompromised mouse, and then you inject T cells and evaluate whether those T cells are able to destroy the tumor. Usually what you see is either the tumor grows out or the tumor never grows. And what we've been able to do is drop down our dose. We made such potent cells that we can drop down our cell dose to the point where you need weeks of time for the T cells to grow. During that time, they're proliferating, they're staying functional, and eventually they catch up to the tumor. That's the peak around 40 days in blue. And then over the next a week and a half, you can see that the tumor melts. So this is unprecedented potency. When you look under the hood, it's unprecedented proliferation and persistence of the cells. I'm going to spend the rest of the presentation telling you how we did that and why we're going to be able to do it again and again and again. So as I mentioned, we use protein design and cell engineering to program how cells function inside patients. It's, you know, our immune cells go to the tumor, naturally they turn off, they make the wrong decisions. We want to reprogram how they make those decisions. We've built our company to be able to go from concept to construct that's ready to go into IND extremely efficiently, and then using these technologies that I'm going to be telling you about, being able to solve the problems in ways that nobody else is able to. And so just the background from our protein design days, we really moved the field from the ability to design proteins with the right structure to the ability to design proteins with completely new biological functions. And this is this is actually how we design proteins these days. We use a similar model to what Dolly and other diffusion-based image generators do, but we use this for designing proteins. And using these underlying protein design capabilities, we've established 5 different technologies that are able to address these large core areas that need to be solved in order to cure cancer. And underneath each one of these technologies, we're able to develop specific assets that tackle very specific biology that is clinically validated. So biology like IL-2, IL-21, IL-12, et cetera. And then the really special thing is that because we're able to develop all of these assets at Outpace, we can combine them into a single cell therapy product and be able to achieve the persistence that we need from one of our technologies and achieve the potency that we need from another and achieve the immune engagement that we need from the other and integrate all of those into the response that we're looking for from our engineered T cells. So this is our lead program. It's targeting mesothelin. It's a CAR T program. We intentionally chose the most validated modality out there for anti-cancer cell therapies. The place where we're really doing the special stuff is on the left there, where we've made the most active mesothelin CAR in the field, powered by our Outspacers technology. We've made the most exciting IL-2 / IL-15. It can actually signal both ways on different cell types in a programmable way that we can use to make the engineered cells better, but also to engage the endogenous immune system. We can regulate the whole thing with dynamically controlled promoters that allow us to both boost safety, but also boost efficacy and counteract mechanisms of dysfunction like exhaustion. Then we also have our EGFR-OP safety switch, which responds to cetuximab, and it works extremely well. That's published. So the first technology, Outspacers, many of you probably appreciate the spacer as the part of the CAR that's between the binder and the transmembrane domain. And the field has appreciated that the length of the synapse is defined by the spacer, and it's really important for how well the CAR functions. We initially designed a bunch of spacers because it's a really crowded space from an IP perspective. We said, hey, we need to have fully human sequences. They need to be derived from extracellular proteins, and they need to have the right biophysical properties to make a good CAR. That's where our protein design and structural biology expertise comes in, and we were able to make this whole panel of spacers that when we actually tested them against different binders, and now we've done more than 10 CARs at Outpace, what we found was that the optimal window of that synapse is much narrower than we previously thought. And it's actually a different length than a TCR synapse, which is kind of surprising. But in effect, what we were able to do is for every single CAR that we've tried to make, we've been able to blow the existing CARs reported in the field out of the water. And one example of that is our lead program from our mesothelin targeting CAR. The one in red is the previous clinical benchmark. You can see it's a not very good CAR. Even at 8 million cells dosed per animal, you can see we're still not even curing the mice with that CAR. If we drop all the way down to 2 million cells with this donor, you can see that we're able to completely ablate those tumors in all the mice that we tested. And again, you can see that hallmark, the tumor keeps on going up before it goes down. So you can see how stringent this assay is. The second technology is our Outsmart technology. And And I think we can all appreciate how important cytokine biology and how our immune system functions. Cytokines are how our cells coordinate a response together. It works great when you've got a viral infection. Doesn't work so great in cancer because the dysfunctional tumor microenvironment leads to dysregulation of cytokine production. So what we wanted to be able to do was replicate the same exquisite level of control that the immune system uses, but in a way that is robust to the dysfunctional tumor microenvironment. So we developed multiple complementary regulation mechanisms to produce the cytokine in the tumor, keep it in the tumor, make sure it stimulates the right cells down the right signaling pathways in the tumor, and then if it leaves the tumor, have it rapidly lose its activity. And we have two different cytokines that we've developed. The one I'm telling you about today is our IL-215 asset. And basically what we've done is to redesign this cytokine. The outside of it still looks very much like IL-2, with the exception that it no longer interacts with CD25, which is one of the key mechanisms that leads to dysfunction, toxicity. And then we've taken that several steps further by being able to retarget it to the cells that we actually want to be stimulating. So you can see on the bottom right there, we take IL-2 in red, which dramatically prefers stimulating Tregs, and we are able to reduce activity on Tregs by several orders of magnitude and increase activity on T effector cells as well as NK cells not shown here. And so that's how we're going to be really tipping the balance of the immune response. And when we take this in vivo, even when we compare it against wild-type IL-2, which is, I think we all appreciate is an even more potent cytokine than IL-15, which is what a lot of the rest of the field is using, we can see that Outsmart IL-215 is even more potent, leading to better responses from these mice, where IL-2 on its own isn't able to control the tumor. When you look at proliferation of these T cells, you can see that we get better proliferation, both CD4s and CD8s that are producing this cytokine. The third technologies are at last promoter that regulates both the expression of the CAR as well as the Outsmart cytokine. This data right here is abstracting away the cytokine, so you're just seeing the benefit that you get from regulating the CAR. The rest of the field usually constitutively expresses the CAR. What we're able to do is use this promoter that keeps a low basal expression of the CAR until you get engagement with the tumor, and then you get massive increase in the CAR expression. And so what that does is, number one, increases the potency of these T cells when they fight the tumor. But number two, it's able to, and you can see that potency in the top right there with the improvement in tumor efficacy across those two different cell doses. And then at the bottom right, what you can actually see is that we have reduced expression of exhaustion markers like PD-1 and TIGIT. We also, not in this slide, have data that shows increased expression of cytokines, which are a great measure of the functionality of these cells. And then the final technology, which I'm not going to go into, you can read it in that paper in the bottom left, is our EGFR opt safety switch. This is one that we think should be in every gene and cell therapy out there. We'd love for people to use it. Please talk to us about that. So I walked you through OPB 101. It's our mesothelin CAR T program. We're going to be taking that into, we're submitting IND next year. Very, very excited about that. What we realized was that we had built an incredible platform of technology assets here that needed to be moved into the clinic. And really all it takes is us generating a new binder against a new target. And so we've, in order to start new programs, we already have more efficacy modules than we can even put into OPB 101. So I haven't even taken you through everything we've done. And so we have two additional programs. One's a TCR, one's a CAR. And we're really excited about diversifying across different modalities, different targets, different indications. We also have a collaboration with Lyle, which is the company that we spun out of, and we have capacity to use these technologies that we're developing in order to contribute to other companies' pipelines as well. So with that, I'll stop there. Please feel free to come up and talk to myself or Lindsay, who's in the audience, or Eric back there, if you have any additional questions. It's been a really fun year of really going from the platform that we had established by the end of last year to creating this pretty incredible pipeline that we've been able to build this year at Outpace, and I'm excited to give you more updates next year. Thank you.